a phosphorylated serine in each monomer. And here the allosteric sites are empty. So if glucose binds, two glucoses bind, one on each side of the dimer, then part of the protein, it's actually the very N-terminus of the protein. It's a huge protein, like 1,000 amino acids per subunit. So that's a big monstrous protein. When glucose binds, there's a conformational change. And so the part of the peptide is that the N-terminus flips out. Right? So then, in response to insulin, okay, we can trigger the pathway that results in the activation of a phosphatase, okay, that will take the phosphates off, and that will inactivate the phosphorylase. Right? So that means you're no longer breaking down glycogen, at least not nearly as much. Does that make sense? Insulin, high blood sugar, glycogen, breakdown, more sugar, more of what we already have, not good. So let's turn it off. Okay?
I have a question.
Yeah.
So if we already have high the blood glucose, then I mean you have insulin. Why do you need both to bind? I guess.
It's just another level of control.
Okay.
It's very finely controlled. Sometimes you might think of it as, well, we really need to turn it off a lot, and each mechanism only works to a certain level of efficiency, so we want to pile them up. But that also allows you to have different levels of control depending on precisely how much glucose there is. So what fraction, what's the Km of this molecule for glucose in the what's the binding affinity in the allosteric site compared to how much there is? What fraction of the molecules is this actually happening for? Right? Not necessarily all of them. Yeah. Other questions? Yeah.
I just want to make sure I understand the slide correctly. After the glucose binds to phosphorylase A, a change occurs where the phosphor groups are available or exposed, and
Yes.
insulin activates one to cleave off the phosphates, and then that turns the phosphorylase A into phosphorylase B.
Correct. Yeah. Yeah. It's the same protein. It just changed its label from A to B when the phosphates came off of it. That's really confusing to me. I think it should just stay. I mean, we should just call it GP, glycogen phosphorylase. Yeah.
So under low glucose concentration, will glucose lose its affinity to phosphorylase B and just fall off? Or does it get phosphorylated and get kicked off?
Probably both. Both, yeah. The glucose goes down in concentration. You can imagine that there's an equilibration, and then it won't rebind if the level doesn't stay high. At the same time, if you phosphorylate it, it'll want to go to the active state, and that may also help kick it off. So probably both. Okay, let's move on. So that was glycogen phosphorylase. So now we're doing glycogen synthase. Okay, so how should we regulate whether we make glycogen? Okay. So here, active and inactive, right? That's the center point of the slide. Now it's active without the phosphates. It's exactly the other way around, right? Which makes sense, it turns out, and you will see that in a subsequent slide. Okay. So in order to inactivate it, we need to phosphorylate it. And in order to phosphorylate it, it turns out that we need two different kinases. And one of them is called CK2, and one of them is called GSK3. And you see here that insulin operates through a pathway to turn off GSK3. And we'll see how shortly. But if insulin turns off GSK3, then glycogen synthase stays active. Is that what we want? Lots of insulin, high blood glucose. Do we want to keep glycogen synthesis on?
Yes.
Yes. Yes, you do. So see, that's one level. It's always one level of the analysis, right? It's like there's something going on in the cell, lots of glucose or not. Therefore, what happens. So you just, one thing is just to know, to be able to recognize which way things have to go, right? Once you know which way things have to go, you've probably got 1/2 to 2/3 of it done. The next part is, well, what's the mechanism by which it goes? Right? But the most important thing is to be able to recognize which way it goes, which just takes a little focus and thought. It is logical. It's just a little complicated. Right? So okay, that makes sense. So let's say it's inactive, and in order to activate it, we need to take the phosphates off. So here's a phosphatase again, PP1. This particular phosphatase is actually bound up in the glycogen particle, and there's kind of more complexity with that we're going to get to. But here's the release of those phosphate groups, and so then we could ask, well, what's going to turn this on, right? What's going to turn on glycogen synthase? Right? Well, insulin wants to turn it on, so here's a pathway here, right? Here, the insulin pathway turned it off. Here, the insulin pathway turns it on. But that makes sense, right? Because for the same logic here, right? If you have high blood sugar, you want to make glycogen. So here it's in the positive sense. If you have low blood sugar, the very last thing you want to do is take the little bit of sugar you got left and throw it into glycogen, where it does nothing for the cell, right? It's a storage particle. So glucagon, epinephrine, low blood sugar, fight or flight response, I need glucose, right? Please give me glucose, because the bear is coming, right? Glucose 6-phosphate and glucose itself, right? Clearly, this is directly related to glucose. This is glucose. So in both cases, turn on, make active synthase. So contribute to the allosteric modulation of PP1 in such a way as to activate it. Right? This will be allosteric effectors of PP1. This will be a phosphatase protein that activates water for attack on these phosphate groups to remove them as phosphate. Right? And somewhere on the other side of the protein will be a glucose binding site. Right? So, does the phosphatase enzyme that activates glycogen synthase have a binding site for glucose? Would it make sense, at least, for it to have a binding site for glucose? Yes, for this reason. Right? Would it make sense for the phosphatase enzyme that activates glycogen synthase by dephosphorylating it? Would it make sense for it to have an allosteric binding site for glucose? And if it does, would you expect that the glucose activates it or inactivates it? That would be an example of a question I would imagine you guys could be able to answer.
Okay. Are there multiple PP1s? So on the previous slide,
there are multiple phosphatases.
Yeah.
The book does not do a good job of distinguishing them. PP2A is a distinctive one for sure.
Mm-hmm.
And then there are PP1s that are in different tissues.
Okay.
So the answer is yes.
Okay.
But I'm going to try to minimize that complexity to the maximum extent I can. This phosphatase is directly bound up in the glycogen particle, and so it can only be involved in glycogen regulation. But the names get really similar. So. All right, so how does GSK3 work? Right. I added this text here just for the lecture, so it may not be if you print out the slide, you may not see it. So GSK3 is a processive enzyme in the sense that it adds multiple phosphate groups. It adds up to 4 of them. And it adds them on glycogen synthase, which has spaced serines to accept the phosphate groups. Right? And so once GSK3 is able to get going, it has an active site that binds ATP and then transfers the gamma phosphate to this serine and then releases ADP and then moves across the protein to get to the next serine.